Okay, so I think we are right on time. And Susanna, all set? Can we start? Okay, so finally we are here, and the day has come when the Plant Genome Journal launches its webinar series. So, hello and welcome to all the participants to the very first webinar of the Plant Genome. I'm your moderator, Nilesh Mishra, the social media editor for the Plant Genome and senior scientific officer working at the International Crop Research Institute for Semi-Arid Tropic in India. Thank you for taking the time to join us today. We are very excited to launch this new series, which will be held every 2 months and will feature authors and exciting research published in the Plant Genome Journal. Please consider sharing this webinar with your colleagues and those who might be interested to learn more in the advancement in the plant genome area. And before we begin, I would like to mention that the questions can be typed in the QA section, which you can see at the downside, the center of the screen. And this webinar will be recorded, and the recording will be emailed to all the attendees in the next several days. So without any further ado, let me introduce our today's speaker, Dr. Susanna Tulpova, who has agreed for this very first webinar in the series and will speak about her research, where for the very first time, a fine genomic organization and tissue-specific expression of ribosomal DNA loci were deciphered in a complex polyploid species. Her research was published in the March 2022 issue of the Plant Genome, and Dr. Tulpova is currently a postdoc at the Center for Plant Structural and Functional Genomics of the Institute of Experimental Botany in Olmo, where her research focuses on optical mapping, including epigenetics, OCT4 nanopore sequencing, and multigenic ribosomal RNA loci in triticale. So with that, let's welcome Dr. Tulpova, and the floor is yours.
Well, thank you, Nilesh, and welcome everybody to today's presentation. Well, first of all, I would like to just thank the Plant Genome Journal and its committee that they have chosen our research and gave me the opportunity to present our work. It's really an honor to be here and to be the very first speaker of the journal's webinar series. So thank you again, and let's move to presentation. So today's topic are the breadwheat ribosomal DNA loci, their structure and transcription. So if I said ribosomal DNA or ribosomal something, the first thing I have in mind are ribosomes, correct? Ribosomes are tiny organelles, and we all know that they are protein-making factories that have been around since the very beginning of life. But the most important and interesting thing at ribosomes is their composition. Why? Because ribosomes are composed of two subunits, large subunit that contains 26 srRNA and 5.8 srRNA, and small subunit that contains 18 srRNA. All three, or these three RNAs, or more precisely saying genes encoding such RNAs, are together to create 45 rDNA loci that are known to be associated with nucleolus organized region. I shouldn't forget on 5 srRNA, that is also important, and it stabilizes key macromolecular interactions. In most plants and animals' genomes, 45 srDNA and 5 srDNA are separated from each other. Their organization is known, but not difficult to hold. The 5S rDNA creates clusters with the repeat units about several hundreds base pairs, so they are tractable to sequencing platforms that producing long reads. On the other hand, 45S rDNA is organized in head-to-tail tandem array. with a repeat unit size varies between 8 and 10 kilobases. And that's why the array size can achieve hundreds to tens or 1,000 kilobases. And this is the reason why they are the major source of gaps in all genome assemblies. And also the reason why the genomic or genetic composition and expression status remain still elusive. Even in case of human assembly, the rDNA array is missing. I'm talking about the well-known human reference called HG38. But on the other hand, there is an exception, and the rDNA array was firstly resolved in the very most recent human assembly that was published let's say something about a month ago in Nature journal. It's telomere to telomere genome assembly of haploid cell line. So far, as I know, also only 1 out of 2 ribosomal arrays was reconstructed in case of Arabidopsis by Simpson co-authors. So as you can see, even in deployed and well-studied genomes, rDNA arrays, are very challenging. And we have chosen bread wheat. Bread wheat is a hexaploid species containing 3 similar subgenomes, A, B, and D. Its genome is really complex and highly repetitive. It achieved nearly 17 gigabase per in size and contains more than 85% of repeats. So it wasn't a big surprise when cytogeneticists in early 19th identified and localized rDNA or rDNA arrays on several chromosomes, chromosomes 1A, 1B, 6B, 5D, and 7D. But still, the regular rDNA arrays are missing in the assembly. So the question is how we can figure that out, how we can resolve rDNA arrays in our assembly. So one opportunity is using the optical mapping. Optical mapping in nanochannels commercialized by Genome. And what is it? Optical mapping is a single molecule mapping of stretched genomic DNA, hundreds of kilobases to megabases in size. Applying specific enzymes, we can create sequence motif maps that you can imagine like a genetic barcode. And such maps provide long-range template for ordering genome assemblies. What I would like to note and emphasize is that not just high molecular weight DNA is essential for optical mapping, but also the presence of recognition sites of enzymes that you want to use. Without recognition site, you can get optical map and you can analyze anything. And we were lucky because we found the recognition size of the most used enzymes called the DLE1 and NTBSPQ1 in our region in 45S rDNA. That site is localized in the coding region of 26S RNA. And as you can see in the picture, it creates really nice regular pattern. So we are lucky and we can use that. So we decided to generate optical maps for all chromosomes that were identified to carry rDNA loci. We applied flow-cytometry to sort them and then generate optical maps. And in all cases, we achieved the molecular coverage higher than 250x, with the average genome map length longer or higher than 600 kilobase pairs. In one case, in case of 5DS, the average genome map length exceeded 5.8 megabase pairs in length, which is amazing. All our optical map aligned to the reference genome with reference genome that was published in 2018 in Science Journal. The green bars are representing the pseudomolecule, the reference sequence, and the blue bars are representing the optical maps. And I think that even if you are not familiar with visualization of optical mapping results, you can clearly see that there is no regular I would like to emphasize, a regular pattern in any of presented pseudomolecules. The one exception, or only exception, is chromosome 1A, because we found at the very end, at the terminus of the short arm of chromosome 1A, some kind of array. However, this array is irregular and it has just short units and fragmented rDNA units that are also, or were also, interspersed by telomeric sequences and other repeats. This is also one of many reasons that we think, or reasons that cause that this array is probably not functional. On the other hand, we didn't find anything in sequences of 1B, 6B, and 5D, just some kind of fragments of rDNA or something like that. But we found regular patterns and regular arrays in all presented optical maps. We also studied the sequence a bit deeper in details, and we found out that at such positions, there are borders between scaffolds in that pseudomolecules that suggesting that there is really missing something and we're not wrong. Moreover, positions of rDNA arrays that we localized, or that we are saying that there is supposed to be rDNA array, corresponds to that that was identified by cytogeneticists. So what can we say about that missing parts? How can we describe that? How big are rDNA units? How many of them are in bread wheat? And how are they distributed? Because we know that there are several chromosomes that are carrying rDNA loci. Can we even answer such questions? And actually the answer is yes, of course. And again, we can use optical mapping, but we can't use optical maps as you saw in previous slides, because such optical maps are kind of a consensus. So that's why we use optical maps single molecule data and apply Python software called IrisView that enables us selecting repeat units in size bins. And using that, we can say that the repeat unit size in 1BS is 9.4, KB 9.5 in case of 6BS, and 8.8 in case of 5DS. The second question was how many 45S rDNAs are in bread wheat. So to investigate that, we used bread wheat whole genome Illumina reads that had 250 base pairs in length and mapped them to the rDNA consensus sequence. Then we calculated the percentage of mapped reads to 18S and 26S, which resulted in nearly 7,000 units in case of 18S and 6,300 and something units in case of 26S. On average, we found out or revealed, estimated, then 6,650 units are present in haploid bread wheat genome, which corresponds to 63 megabase pairs in length. And the last question was, how are 45S rDNA units distributed? To answer that, we need chromosome or needed specific chromosome rDNA units. To reconstruct chromosome-specific rDNA units, we used published read chromosome reads that were published in 2014 and repeat explorer pipeline. The entire architecture of the repeat explorer is shown in this graph, but don't worry, I won't go in details because it would be for another presentation. So the most important thing is that the pipeline includes graph-based clustering to identify groups of reads derived from repetitive elements and is able to characterize them with respect to the genome. Using that, we got the chromosome-specific rDNA units. We also compare them between each other as well with the consensus rDNA sequence, and we revealed that there is a high sequence diversity in intergenic spacers and also in the internal transcript spacers, but that was not a big surprise. What was a surprise was the fact that we also identified SNPs encoding region of 26S And such SNPs also became diagnostic ones. Why? I didn't emphasize that, but our reconstructed units were also validated by whole genome reads that had 470 base pairs in length. Based on that, we were also able to determine SNP haplotypes specific for particular chromosomes. And thanks to that, we can say that 1/3 of all identified or estimated rDNA units originated from 1bs chromosome, nearly 2/3 originated from 6bs, and the rest of them originated from the other loci, when the biggest portion belongs to 5ds. So we know the distribution, and the logic question could be, what is the transcription activity of individual loci? To be honest, we didn't know much. I mean, we didn't know what to expect. So that's why we isolated, sequenced, and analyzed total RNA from 5 different plant tissue at different development stages. So just to remind you, when we are talking about rDNA, 6bs locus dominates. But when you check RNA-seq data and RNA-seq results, the situation is completely different. Suddenly, 2/3 of transcripts originated from 1bs, and just 1/3 originated in 6bs. So that means that the amount of 1bs and 6bs transcripts is not proportional to the locus size. Then you can see it in the table, proportion of 6bs transcripts decreases during the development. And finally, it is not written in the table, but transcription of 5ds and possibly 1as unit was approximately about 1%, and showed the same developmental trends as that of 6bs locus. So to clarify the mechanisms controlling expression of the RNA of our interest, we analyze cytosine methylation levels in rDNA loci using data from bisulfite sequencing from LEAF that were published again in 2018. And I think that it's clearly see, at least at the context of CHG, that 5ds was silenced preferentially by cytosine methylation in such context, and the rest two are similar or almost the same. And then we also revealed a significant difference and significant increase in CpG methylation in case of 6bs. Putting everything together, we can also describe not the evolution, but we can discuss our results at this context, because the bread wheat is, as I said, allohexaplate genome, and it is a result of 2 independent hybridization events. That's why we have 3 subgenomes, and we found rDNA arrays in all of them, but at different conditions. In case of A genome, we found an rDNA array, we can call that array, at the terminus of the short arm of chromosome A, but that array is showing low homogeneity with short basic units. And if you can remember, those units were interspersed by telomeric repeats. On the other hand, B genome dominates in all aspects of our research. We found 2 arrays showing high homogeneity and long basic units. We can say that both arrays are active, and we also revealed inverse correlation of NOR activity to locus size. And finally, the D genome, we found an array there with high homogeneity, but with short basic units. And what was actually a surprise for me, we revealed increased CHG methylation, and which suggesting epigenetic silencing. So I think I can give you many many things to think about, but it's time to summarize everything and make some conclusion. So by combining cytogenetic approaches as well as bioinformatics and several data sets, we have proposed a powerful approach to study individual 45S rDNA loci in complex polyploid genomes. We reconstructed chromosome-specific rDNA units that became a stepping stone for locus-specific analysis. We revealed locus and tissue-specific expression of RNA genes or ribosomal RNA genes, sorry, and also describe genetic and epigenetic mechanisms of our RNA dosage control. Last but not least, our data indicate different evolutionary histories of rDNA loci in the 3 subgenome of bread wheat. So that's all from my side. I hope you enjoyed the presentation, and thank you for your attention. If you have any question, I would be happy to answer. Thank you again.
Thank you, Susanna, for that very being on very time, and also for sharing your research for the benefit of the participants. With that, now we are on in our Q&A session. So I request all the participants, if they have any questions, please do type in the Q&A section of this. So there is one comment, I would say, rather than a question, from Alice Kovarik. Hello, isn't it a bit worrying that the information about rDNA in assembled genome is actually coming from crippled and particularly non-functional loci? In other words, functional loci are not present in assemblies. Do you have an idea how to deal with this? And thanks for a wonderful talk, Susanna. So that's the message. So you'd like to respond?
Okay. I will have to go through it. So function, yes, I think that it's the matter of I think, in my opinion, it's the matter of the units themselves. I mean, their status, like the non-functional array is irregular. So this is easier for assemblers to assemble the sequence, because there are many differences, and that's why the data won't collapse in one exact position. That's why the regular pattern are not there, are not the assemblies, because when the assembler is trying to assemble the sequence, it's just sorry for the expression, stupid software, and it doesn't know that the repeat is supposed to be 1 by 1, and because the pattern is the same, so it just easily collapses at one position. That's why the irregular is likely to be in every assemblies rather than the regular ones. Is that clear? Hope so. I'm not sure.
Yeah, I do hope that that answers the query. And yeah, so there is another comment from Dr. Henry. He says, nice work and wonderful talk. Thank you. So that's an appreciation for you. Let's see if we have any other questions. So, which means that your presentation was very lucid and clear. So that could be only 2 reasons. So it seems everywhere that things are very clear to all the participants. No more further questions. Okay, so then there's a response from Alice again. I feel that the problem is not just assemblers, but regular units may be missing from long reads. Yeah, that's more like a...
That's... Yeah, agree. Thank you.
So I don't see any more questions. So maybe I can ask one if that's fine with you, Susanna.
Okay, no problem.
Maybe very generic one. So is this approach applicable to other genomes as well? Like you work for the cereals and some other legumes crops as well? How do you see that?
Actually, yes, it is. The essential thing is that, actually, I have mentioned it already. The essential is to have the recognition site of the enzyme used for generation of the optical map. If you have the recognition site, you can analyze that. So yes, it is applicable. We also applied our approach to, for example, to barley, and also try to apply it on Vicia faba, but we weren't successful there. But we got really nice results in case of the barley.
We have a comment again. Okay, so that's more for me. Thank you, Nilesh, for excellent organization of the webinar. Thank you very much, Alice, and thanks to all the team members from the Plant Genome who has put together this webinar series and also the very first webinar in the series. So thanks to all of them. Yeah. So Matt has shared your research article, the link to your research article here for the benefit of participants. If they would like to read more about the research article and the work that you have presented here, they can for sure go through the link. And I'm sure if at this moment, very moment, if the participant doesn't have any questions and if they come up anything later stage also, they can for sure reach out to you, Susanna. I think that should be fine, right?
Yep.
Okay. Well, I think we are reaching to the end of our very first webinar, because I don't see any further questions or any comments here in the Q&A and the chat box. So let me thank you once again, Susanna, for sharing your time and also your excellent work that you published in the Plant Genome Journal. Thank you very much.
It was a pleasure. Thank you.
Yeah. So yeah, back to, I think we are at the end of the webinar. So let me again thanks to all the participants who have joined. And again, as a reminder, the recording will be made available and sent to all the attendees. Thank you again for giving us your time, and have a good day. Goodbye and bye-bye. Thank you very much.
Bye. Bye.